Hello students, this is Professor R. M. Mandrakar from JSS Arts, Science and Commerce College, Gokak. Today, in this part 3 of plant tissue culture, we'll be studying the basic steps involved in the technique. Now, this is a flowchart of the plant tissue culture so that you'll get used to what all the different steps that are involved in this technique. First, preparation of medium. You know that the plant medium plays a very important role. So how the medium is prepared, and after the medium is prepared, it will be sterilized in autoclave, then it is poured in bottles, then it is again sealed and re-sterilized, and then it is poured into the petri dishes and allowed for setting, and now your medium will be ready for inoculation of the plant tissue. Now, preparation of the plant. First, you have to select a mother plant, that is the plant of interest. Then from this, a part of that plant is cut. So this piece of plant extracted from the mother plant, we call it as an explant. Then this explant has to be washed because it will be covered with dirt, foreign pollen grains, chemicals. All this has to be cleaned, and it is again re-washed and sterilized, and then it is shifted to the lamellar airflow station, and then it is again sterilized with the sodium hypochlorite, and it is washed 3 to 4 times, and now the explant is ready for inoculation. So now the explant will be introduced in the nutritive medium, then it is shifted to the growth rooms where it is maintained at 15 to 25 degrees Celsius, and then after 3 to 4 weeks we will be seeing a bunch of in vitro shoots. Then these shoots are separated and then they are transferred into the rooting solution in the growth rooms, and then after 1 to 3 weeks the plants are then transferred to the greenhouse for hardening. First, let us study about the preparation of the culture medium. Preparation of culture medium for plant tissue culture is one of the most important part. One should take into account to place the cell or the plant tissue in a media which contains the same nutrient that it would require when growing in nature or in the wild. So, if you can maintain a perfect environment while supplying the plant media and culture media with proper nutrients, you should have a successful tissue culture on your hands. A convenient method for preparation of culture media is to make concentrated stock solutions which can be immediately diluted to preferred concentration before use. For the establishment of new protocol for a specific purpose in tissue culture, a suitable medium is better formulated by testing the individual addition of a series of concentration of a given compound to a universal basal medium such as the MS medium, which is the Mursinge-Skoogs medium, or the LS medium, that is the Linzmeyer-Skoogs medium, or B5 medium, that is Gambox medium. The following steps outline the proper preparation of the media for tissue culture. Now you can take any one of those above mentioned media in appropriate amount. If you are mixing for 1 liter medium, then fill a beaker with 800 ml distilled water. Slowly begin adding the powdered medium into the beaker. Then add 30 grams sucrose, set the pH at 5.8, add agar to the beaker, that is about 8 grams, then add hormones if you are using it, and add 200 ml of distilled water. Then this is transferred for the autoclave purpose. That is, prevention of contamination of the tissue culture medium is important for the whole process of the plant propagation and helps to decrease the spread of plant parasites. Sterilization of media is roughly achieved by autoclaving at the temperature ranging from 115 degrees Celsius to 135 degrees Celsius. This sterilization is important to avoid the bacterial contamination. Advantage of autoclaving are the method is quick and simple, whereas disadvantages are the media pH may change and some components may decompose or lose their effective. The next step is pouring this sterilized medium into bottles. The sterilized medium is then removed from the autoclave and then it is poured into the culture bottles and each bottle is labeled. Then the bottles are again capped and sealed with the paraffin tape and then it is again sterilized. The culture media is then poured into the petri dishes and it is transferred to a sterile environment and stored. The media should be left in a sterile environment where it is monitored until it can be used for tissue culture later. So thus now your medium is ready for inoculation. The selection of the mother plant. Now, when you select a mother plant, you should always take into consideration the beneficial features. That is, here you can see this is a strawberry plant. It is showing healthy growth. It is disease free and it is giving large quantities of strawberries. So the output, the yield is more. So all these criteria that the plant should be healthy, it should be disease free, it should give a high yield and it should be resistant, it should be drought resistant. So all these important characters are taken into consideration when you are selecting a mother plant. After the specific mother plant is selected, then a part of the mother plant is cut out. Now here part of the stem of the strawberry is cut out. So this part of the stem of the strawberry, now it acts as an explant. Now this explant is then washed so that whatever the dirt is there on that, whatever the foreign pollen grains that are present on that, so whatever the bacteria and other viral particles that are present on that, all these are removed. It is again re-washed and the surface is sterilized. Of the explant, it is then shifted to the lamellar airflow station. Now this is an image showing you the laminar airflow station. Now this laminar airflow station has got this following three chambers. That is, the first chamber is the inlet chamber. The second is the compression chamber and this third is the working chamber. Now the propeller in the compressor chamber, it generates a stream of air that is sucked through the pre-filter. Now this inlet has got a filter here, so we call this as a pre-filter. So whatever the air that is coming here, it is filtered here. And then this air is then passed through this HEPA filter. Now this HEPA filters are highly efficient filters. They typically capture over 99.5% of all particulate matters. They are made up of either plastic or PET or fiberglass, and they can capture things like pollen, virus, bacteria, molds, and particulate matters of 2.5. The explant is treated with the disinfectant in this working chamber. Now here you can see this is another image of this laminar airflow station. Now here you can see this is the inlet here, this is the compression zone, and this is the working zone. So whatever the air that is moving here, it is fully sterilized, and here we have this ultraviolet light, which again kills whatever the bacteria that are present here. And you can see this man is working with the tissue culture processes. That is, first, once the explant is transferred to this laminar airflow station, it is again sterilized by this sodium hypochlorite, and after this treatment the explant is washed thrice with double distilled water to remove the chemical remnants. The explants are then inoculated on the sterilized medium. The inoculation takes place in the inoculation room on the laminar airflow station, which maintains a continuous air current which keeps the bench without the risk of contamination. The expert technicians in the laboratory do the inoculation. Then after the inoculation process is over, the culture is then moved into these growth rooms. Now this is a growth room. It is a well ventilated, air conditioned, and 1000 clean air is maintained in this growth room. So inoculated cultures are then shifted into these growth rooms, where a typical temperature of 15 to 20 degrees Celsius is maintained with the help of air conditioners, and class 1000 clean air is maintained. The artificial lighting arrangement is also made for the growth of the plants. Now this is the inner view of that growth chamber, where you can see these racks. All these are totally sterilized. It is 0B condition here, so that the contamination doesn't occur in the culture medium. Then after some 3 to 6 weeks, the inoculated explant shows growth in multiple shoots. These shoots are then transplanted on the subculture or multiplication medium for further growth of the plants. Now here you can see the explant has produced so many numerous number of these shoots here. Now these shoots are separated into separate culture chambers. So this we call it as subculture. For subculture, separate growth medium is prepared. Several subculture cycles are done in the laboratory for mass production from the explant. The next is separation of in vitro shoots and rooting. Now once the shoot system is developed, then we induce the root system by changing the auxin and cytokinin ratio. And then once the plant, okay, now here you can see this is the shoot system developed here, but the root system has not been developed, then it will be shifted to another culture by varying the auxin and cytokinin ratios. Now these are done by trial and error methods, and then we'll see the rooting takes place. So once the rooting and the shooting occurs, then this is a perfect medium for the culture of these subcultures. So that is the main reason why when you're doing subcultures, we make a large number of these culture tubes, and in each of that we keep a slight variation in the nutritive medium, so that we'll come to know at which specific ratio of oxygen and cytokinin the rooting occurs in these plants. Then after this technique, once the shoots are fully developed, the plants are then transferred into the small crucibles containing the soft soil, and then they're transferred to the greenhouse for hardening. Rooted shoots are removed from the laboratory and are transferred to the greenhouse for hardening. In the hardening procedure, the plants are first kept in humidity chamber for acclimatization, and then they are transferred into the greenhouse. So here the actually hard tissues, that is the sclerenchyma tissues, the vascular tissues are developed, and now the plant is getting used to the outer environment, and so from one small piece of the explant we can get so many numbers of those plants. So that is the benefit of this tissue culture, and whatever the plants you are seeing here, they're all the clones. That is, they have the same characters, the same morphological features, the same output of that of the mother plant that you had selected for tissue culture. So thus, to conclude this topic, tissue culture activity is taking a shape of an industry, as many farmers are planting tissue culture grown plantlets. Agro traders are buying and selling tissue culture grown plantlets, while some are exporting either the plantlets or the producers of the plants grown by tissue culture, especially varieties of flowers like roses, anthuriums and zerberas are enjoying high profit. Tissue culture is the greatest advancement in plant breeding. Agriculture and horticulture industry should take full advantage of this technique in reaching the great heights at national and international level. In India, tissue culture is rapidly becoming a commercial method for propagating new and rare species and difficult to propagate plants. For a few research laboratories several years ago, a whole new industry is emerging. Currently, the demand for micropropagation plants is greater than the supply with some plants. Some growers specialize in only the micropropagation of plantlets, leaving the grow-on, that is, the hardening activities, to the other. Many growers are integrating a tissue culture laboratory into their overall operation. Hence, tissue culture is one of the most important techniques of obtaining all the good varieties of plants. So that's why we say tissue culture has played a great role in improvement of plants. Let it be a crop plant, let it be ornamental plants, let it be flowering plants. So with this, I conclude this topic on this tissue culture. Thank you. Please take care of your health. Be safe. Whenever you go out, please wear mask, use hand sanitizers, and as you know, now the second wave has started. So please, please, please take care of your health and be safe. Once again, thank you.